Hello everyone, my name is Claudia Rivas, and today I will be talking to you about a study called PIKE3CA mutation confirms resistance to chemotherapy on triple-negative breast cancer by inhibiting apoptosis and activating the PI3K-AKT-mTOR signaling pathway. This is a relatively recent study. It was actually published in March 2021 in the journal Annals of Translational Medicine. I'd like to thank Dr. Trubal for giving me the opportunity to present this to you today. The research questions that the authors were trying to answer were the following. They were trying to find out if modification to the expression level or mutations to the PIKE3 gene lead to any of the following. They wanted to know if there was any enhanced cell proliferation, if it inhibited apoptosis after chemotherapy treatment, whether there was an enhanced migratory ability of TNBC cells, the activation of the PAM signaling pathway, and whether there was a negative prognosis of TNBC patients. A little bit of background information. TNBC stands for triple-negative breast cancer, and it's characterized when estrogen receptor, progesterone receptors, and human epidermal growth factors are lacking in these cells. TNBC accounts for 10 to 20% of breast cancers, and it is characterized to have poor prognosis, where only 20% of the patients achieve PCR. PCR is basically the disappearance of the cancerous tumors after chemotherapy treatment. So given the poor prognosis, we can conclude that the development of chemotherapy resistance is pretty common in this type of cancer. It's treated via NAC, also known as neoadjuvant chemotherapy, and epirubicin is one of the drugs used in chemotherapy, and it induces apoptosis and inhibits DNA replication. The PIKE3CA gene is an oncogene that codes for the P110A protein subunit which regulates cell proliferation and apoptosis. This gene codes for the P110A alpha subunit, which phosphorylates PIP2 and converts it to PIP3. The PIP3 then activates downstream signaling pathways like the PAM pathway. The PAM pathway activation is common when the PIK3CA gene is mutated. So there's higher activation of this pathway. There's also a noticeable reduced dependence of growth factors that lead to uncontrolled cell growth. Common mutations are in the regions E545K and H1047R. And there is a noticeable poor response to chemotherapy when these mutations are recorded. The PAM pathway involved regulators. So the PI3K-AKT-mTOR pathway, also known as, I'll be referring to it as a PAM pathway, regulates cell proliferation. So basically the PI3K is an upstream protein in the gene sequence which phosphorylates AKT, which then phosphorylates mTOR, and together they all regulate cell proliferation, survival, and death. Some noticeable or important regulators of the pathway are the P70S6 regulator, the 4E-BPI regulator, and P10, which is a tumor suppressor negative modulator of PAM. Apoptosis regulators that we will be looking at further are the ZAP, BCL2, which are inhibitors of apoptosis, and the caspase-3, which is a promoter of apoptosis. So experimental context that we will be reviewing. We will be looking at in vitro, in vivo, and clinical analysis of the mutations. and this will be analyzed looking at gene and protein expressions, phenotype analysis, and clinical analysis. Important cell lines that we will be looking at are MDAMB231 and MDMB468, which is the Pike3CA wild type. The last one I was reading, the 468. The tested samples are non-control groups, I will be referring to them sometimes, are when the gene is overexpressed, also known as OE, when the E545K region is mutated, I will be referring to it as you see here in parentheses, and when the H1047R region are mutated. And discrepancies of their findings and of their data will be discussed at the end. So I just don't want to confuse any of you when I'm reviewing the data and the results. Okay, so we will be looking at in vitro cell analysis. So both cell lines, the MB231 and 468 cell lines were transfected with the mutations we discussed, or the overexpression of the cell via lentivirus transfection, and their optimal density was measured, and it gave us this chart where the absorbance at that given wavelength were measured. This was all to analyze whether the modified gene promoted cell proliferation. This analysis was known as, it was used to test their cell viability via CCK8 assay. And we found that the overexpressed and both of the mutated regions of the cells grew faster compared to the control in both cell lines. That means in the 231 and 468. And we can see that in these graphs right here. We can see that the overexpressed, which is in the red, the purple, which is the H1047R mutated cells, and the E545K cells were all proliferated more than our control cell, which is seen in the blue. And the same was noticed in the 468 cell line, where our control showed the least amount of cell proliferation. The conclusions the authors drew from this were that the cells grow faster when the Pike3CA gene is either overexpressed or mutated. They also analyze the cell cycle via flow cytometry, and we know we're used to seeing flow cytometry as like a graph with 4 quadrants, and we will see that later, but this time they showed it in this type of graph. And they basically concluded that the 231 cells show the mutations and modifications both increase significantly in the OE sample. So we can see that in this bar graph right here, with the cell proliferation of the OE is significantly larger than the control and also the mutated cells. There was a slight increase in cell proliferation in both mutated, but it wasn't statistically significant, so that's something that we do need to note. In the 468 cells, however, there was no significant, because there was a difference, but it wasn't statistically significant in cell cycle between the control and the sample groups. So in conclusion, based on this data, they concluded that modifications do not improve cell proliferation ability of TNBC cells in vitro. That's very important to note. They also did an apoptosis analysis via flow cytometry, and they basically wanted to know whether these gene modifications inhibited apoptosis. So an important thing to note about the flow cytometry is that we're looking at this quadrant right here in all of the graphs. This quadrant specifically looks at early apoptotic cells. And we can notice that in the 231, the proportion of early apoptotic cells compared to the control groups, there was a decrease in these apoptotic cells for all modified pica genes. So the control had a higher number of early apoptotic cells compared to when the gene was overexpressed, mutated at this region or at this region. The same was noted for the 468 cell line, where the early apoptotic cells was also higher in the control group compared to the overexpressed genes or both of the mutated gene samples. So from this data, they derived to the conclusion that the PIK3CA modifications, both the overexpression and both mutations, inhibited TNBC cell apoptosis. That should be TNBC. And I will move this so you guys can see this. They basically also represented the same data in a bar graph, so it's easier to visualize because I know it's not as easy to look at or to visualize here. So they represented it here where you can see that obviously the control has a higher quantity or percentage of early apoptotic cells, and that's also noted in it's noted in both the 231 cell line and the 468 cell line. They also wanted to know what are the effects of the PIK3CA modifications on migration and invasion of TNBC cells. So this has to do a lot with metastasis of the cancerous cells. They did this via transform migration assay, and they found that the 231 cell line samples, all the non-control groups, this meaning the overexpressed group and the regions of mutation, both showed enhanced aggressiveness. And we can see this here where the darker areas show the TNBC cells, and the overexpress is clearly higher than the control group. And the same for both mutations, the E545K and the H1047R. So the darker circles indicate migration of the cancerous cells, and they're clearly higher in all groups. However, for the 468 cells, the overexpressed and the H1047R mutated samples are more aggressive compared to the control and the E545K. So we can see that here, where the darker areas of these cells obviously represent a higher level of migration, whereas in this group, the E545K is not as aggressive, we can say. So from this data, they derived that the pica, I keep calling it pica, is a PIK3CA modifications provide an enhanced migratory phenotype in TNBC cells. Also, this was backed up, or this data was modified to bar graphs, where again we can see that the controls in both cell lines, the number of invaded cells is significantly lower than all tested sample groups, except this one right here where it was not statistically significant, and that's something that we will discuss a little further down the line. They did test the modifications of the gene and its effect on drug resistance. So they did this by doing a CCK8 assay and an IC50 curve. So this is basically where they tested drug concentrations, specifically of epirubicin that we talked about earlier. So we noticed that As drug concentration increased for both cell lines, the cell viability decreased. So this is fairly expected, we can say, but we do notice that the control group had a significantly less cell viability compared to the overexpressed gene and when the gene was mutated. Another way to explain this is that the control group had lower levels of surviving cancerous cells, if that makes sense. And this is seen in both the 231 and 468 cell lines. From this data, they concluded that the PIKE3CA modified groups are less sensitive to chemotherapy. From this, they wanted to see the effects on apoptosis after the chemotherapy treatment was introduced. And they did this again by flow cytometry. And they found that in the 231 cell line, the proportion of early apoptotic cells decreased among all gene modified groups, including the overexpressed and the mutated. And again, we can see it here where this quadrant right here, which is the one we're looking at, is the early apoptotic cells. The amount of them is significantly higher in the control group versus the mutated groups and the overexpressed group. However, in the 468 cell line, there was decreased proportion of early apoptotic cells in the overexpressed and E545K mutation groups, and an increased proportion in H1047R group. That means that there was an increase in apoptosis in the H1047R group. So that can be, again, we will discuss this a little further, how this might affect not our results, but our conclusions. And we can see that here, where in the overexpressed group and in this H1047 mutation, there was significantly less cells in the early apoptotic stage, compared to the H10 group. It was this. Okay, so this one and this one are the ones with less early apoptotic cells. I said that wrong. Compared to the control, and then this one had an increase in apoptotic cells. And again, this can be viewed in this bar graph where the 231 cell line did have significantly less early apoptotic cells, and the numbers that we derived from the 468 flow cytometry did conclude that the rise in apoptotic cells in both of the mutated genes were not statistically significant. So this is again something that we will look at a little further. Again, they wanted to note the effect on apoptotic protein expression after the epirubicin treatment or chemotherapy. And they did this again by a Western blot, and they only did this on the 231 cells. They did find that all three non-control groups, or the overexpressed and both mutated regions, did show an upregulation of the CAP and BCL2 proteins, which are apoptosis inhibitors. So as we can see here, the CAP, which is an apoptosis inhibitor, was upregulated in the overexpressed gene after the epirubicin treatment. And we can see that for the E545K mutation before and after chemotherapy, and as well as in the H1047R mutated gene before and after epirubicin. And the same can be said for the BCL experiment, where the BCL was overexpressed after chemotherapy treatment. And we can see this by the darker areas shown here. So we're gonna be looking at these two again, a higher expression of BCL2 after treatment and E545K. And again here. And again, if we think about this, they derived to the conclusion that the PIK3CA modifications do inhibit apoptosis caused by epirubicin. The experiment was then taking a step further and tested using in vitro cells cultured in mice, where they took 64 total mice. They divided them into 4 groups, 16 mice each. One group was for the control, another one for the overexpressed PIK3CA gene. The other one was for the mutation at the E545K region. And the last one was where the PIK3CA gene was mutated at the H1047R region. So basically they wanted to know if the tumor grew faster in these modified groups after chemotherapy treatment. So first they subcutaneously injected the 231 cells into these mice, and they allowed them to grow tumors in culture in vivo for 10 days. Then they were treated with epirubicin after the tumor grew to 250 millimeters squared. Cubed, that's cubed. And they did find that the volume of tumors in all PIK3CA modified groups increased significantly faster even after chemotherapy treatment. So again, keep in mind that all of these are after the chemotherapy treatment. And we did see that in the control group, the tumor volume was significantly less in the control group compared to the when the gene was overexpressed or mutated at either of the mentioned regions. They also tested apoptotic protein expression in these tumors via immunohistochemistry. So basically they took the tumors that grew in these rats, and they sliced the tumors to be able to look at the cells. And after staining, we were able to look at the expression of caspase-3, which is an apoptosis promoter, and a ZAP protein, which is an apoptosis inhibitor. And we did conclude that in all 3 groups, so the overexpressed and both mutated regions, the caspase was downregulated. In the control, you can see the darker red areas are where the caspase is expressed, and we no longer see that in either of the control groups. However, the ZAP protein was upregulated in the 3 test groups. And if we think about that, the apoptosis promoter, which is the caspase-3, was downregulated. So basically, overactivation or mutation of the PIK3 gene did cause the apoptosis promoter to be downregulated. So apoptosis wasn't seen as frequently, we can say, because its caspase isn't present. And contrary to the ZAP, which is an apoptosis inhibitor, which is not something we want in cancer cells. We want those cancer cells to undergo apoptosis. So the fact that the ZAP was upregulated, it's not a good thing when speaking in terms of cancer, because that means apoptosis is being inhibited. So to conclude, this part of the experiment, in vivo and in vitro data suggests the pica-3 modifications induce chemotherapy resistance in TNBC cells by inhibiting apoptosis. Another way we can say this is that when the PIK3CA gene is overexpressed or mutated, we're inhibiting apoptosis. Therefore, we can see chemotherapy resistance because, like we mentioned before, the epirubicin does promote apoptosis, if that makes sense. So then the authors wanted to know what molecules are responsible for the apoptosis inhibitory characteristic of the PIK3CA modified TNBC cells. Basically, they wanted to know the molecular mechanism by which apoptosis was inhibited, and they did this by running a quantitative expression of the PAM proteins via Western blot. And they did find that in the 231 cells, the PIK3CA modifications increased expression of AKT, phosphorylated AKT, mTOR, and phosphorylated mTOR in all non-control groups, or in all groups in which the gene was modified. And we can see this again here, where after treatment, the proteins mentioned here were upregulated. So you can see after treatment, the control group, the phosphorylated AKT, was expressed higher, is seen at a higher concentration, as well as here for the E545K sample before and after treatment. And the only discrepancy I see is here. That might have been an error that we will talk about later. And the same can be said for the AKT, where expression of the protein is higher after chemotherapy treatment. And this part right here, I don't know if I mentioned it in the earlier Western blot, this is used as a control of beta-actin. But again, here the mTOR and phosphorylated mTOR are overexpressed or upregulated in after chemotherapy treatment. You can see here based on the size of these bands. This was darker, meaning it was more highly expressed. So what they derive from this information is that the overexpression or mutation of the PIK3CA gene contribute to activation of the PAM pathway. So because, so in other words, this mutation led to an increase, not overexpression, but an increased expression of proteins linked to the PAM pathway. Oh, sorry. Then they wanted to know how the modifications to this gene affected PAM protein and mRNA expression on the mice tumorous cells. So they took it a step further and studied it in vivo. And again, they did this by the quantitative expression of PAM proteins of mice tumors via Western blot. So they basically took cells from these tumors, and again, they ran a Western blot assay. And they found that the P110 alpha subunit, the phosphorylated AKT, mTOR, P4E-BPI, and the phosphorylated P70S6K were all upregulated in the modified tumors. However, PTEN, which if we remember is a tumor suppressor, was downregulated, and that is not something we want to see in cancer cells. We do not want to see tumor suppressors be downregulated. So again, we can see it here, where if we remember this protein right here is what the gene codes for. It was upregulated compared to the control group in all 3 tested samples, as well as the phosphorylated AKT, and the PTEN was downregulated. It's a little hard to see on the bands, but we can see it in this bar graph right here, and I'll get to it in a second. The mTOR was also upregulated, as well as the phosphorylated P70S6 kinase. And the P4EBPI, and I'll just move over. Oh, no, go back. So again, this was the PAM proteins at the expression using the Western blot assay. But then they took it a step further and they analyzed it at the mRNA expression level. And they did this by reverse transcriptase PCR assay, using an RT-PCR assay, and they found that, again, these results were confirmed. So again, we can see that the P110A subunit is upregulated in the overexpressed and mutated samples. They did find that the expression of, not expression, but mRNA levels of AKT were higher than the control group, but they were not statistically significant. Again, they found that the mTOR was upregulated in all 3 tested samples, but they did find that the PTEN mRNA levels compared to the control were downregulated for the mutated samples, but was not statistically significant for the overexpressed sample. So that's something that they can look back into. We'll discuss that a little further. They did find that the 4E-BP1 protein or regulator was upregulated in all 3 samples compared to the control group. They took it again a step further, and they wanted to look at the expression of PAM proteins of mice tumors via immunohistochemistry assay. So again, they took cells from these mice tumors and they tested the PAM proteins. And they did find that the mutations upregulated PAM proteins and downgraded PTEN expression. And then they also found that the overexpressed samples showed high expression of the PTEN protein compared to the mutations. We can see that here. So the PTEN, you see the control group, there's high levels of PTEN, and it doesn't really go down on the overexpressed sample, but there is significantly decreased PTEN expression in the mutated genes. They did find that for both of the mutated samples, there was a high AKT and mTOR proteins, but low PTEN levels. And we can see that here. So compared to the control, AKT is highly expressed in the mutated regions, and so is mTOR right here. See them in the red samples. But now if you look at the PTEN, which again is the tumor suppressor in the mutated samples, there was a decrease in this tumor suppressor. So that's something that they took into consideration. And they ran a clinical pathological analysis of this, of mutations to this gene. So basically, they took 50 patients with a triple negative breast cancer, and all of these 50 patients were previously diagnosed with invasive cancer that they found, and they found that 11 out of these 50 had pica mutations. And based on their analysis, they found that those with the PIK3 mutations had a 91 versus 7.7, both relapse and death. So more negative outcomes, meaning that compared to those that didn't have, those patients that did have TNBC but didn't have PIK3 mutations, those with PIK3 mutations had a higher probability of either relapsing or passing away due to their illness. Based on this data, they concluded that mutations to this gene might be associated with either relapse or death, or both. They did take, they ran another immunohistochemistry assay from tissue of this tumorous, tumorous patients. They found that they wanted to test the PAM proteins and their apoptosis regulators via, again, the immunohistochemistry assay, and they found that AKT and mTOR increased and mutated, and notice that it says mutated pica tumors. That doesn't mean that the overexpression showed the same, but again, this wasn't tested. It was only for the regions that were mutated, because again, this is, we can say, an in vivo test, because they didn't find any, or the patients that they did study using this technique didn't show levels of overexpressed PIK3CA gene. They also found that the PTEN and BCL levels decreased in the H1047R group and increased in the E545K group. And they found no significant difference between ZAP or caspase-3 between the three groups. So we can work backwards or look at this. There was no significant difference in either this one group right here. As we can see, the levels of the regulator are basically the same throughout all three samples of control and the mutated genes, and the same for caspase, where the levels of caspase-3 is basically the same between the control and the mutated samples. And then for P10, if we look at P10 and BCL2 right here, they decreased in compared to the control. So here we're looking at the red cells. P10 decreased from control comparative to the 1047R, and the E545K also show, I wouldn't say they decreased. I think they concluded that these two were about the same. And for BCL2, the same. So there's decreased level of BCL2 in the H1047 mutated group, but they did conclude that the BCL2 was the same, if not increased. I'm gonna say, no, this definitely increased. The BCL2, oh no, go back. Okay, the BCL2 group protein increase in the E545K sample. Again, mTOR and AK7 are correlated to the PAM pathway, and they noticed that there was increased levels of both proteins in the mutated versions. It's not as noticeable here, but you can definitely see it for the mTOR protein. So the authors arrived to the following conclusions based on their tumor volume analysis in mice. Did they conclude that PIK3CA mutations or overexpression led to cell proliferation or promoted growth of the cancerous cells? They also concluded that there was an inhibition of apoptosis even after chemotherapy treatment, and this was based on their apoptosis flow cytometry analysis on the test previously mentioned. They, based on their transpo assays, they concluded that there was an enhanced migratory ability of TNBC cells, and due to their Western blots and immunohistochemistry assays, they concluded that the modified gene samples activated the PAM signaling pathway. Based on their clinical analysis, they associated these gene modifications, not modifications, specifically mutated, they associated these with relapse and death of TNBC patients. Discrepancies and shortcomings. Despite being a fairly thorough paper where they included in vitro, in vivo, and clinical analysis, there was a few, I would say, shortcomings, and just, I'd say they dramatized their data to fit what they wanted to convey. So they reached the conclusions that the PIK3CA mutations led to proliferation of cells, but in vivo data showed otherwise. This was noted when in figure 1C, only the 231 overexpressed sample showed a significant measurement of cell proliferation. I think this is a very erroneous conclusion, especially considering when only one of your samples showed cell proliferation. I don't think that's correct. You shouldn't derive to such conclusion based on evidence or data collection from one sample when you tested several. They also concluded that the PIK3CA modifications led to enhanced migratory abilities, even when the 468E545K mutated cells showed insignificant migration. Again, you can't derive to these conclusions when one of your tested samples did not convey the conclusion you arrived to, and this was seen in figure 1G. Also concluded that the modifications inhibited apoptosis after chemotherapy, even when the recorded data decrease in 468E545K group was marked as not significant, and there was a slight increase in apoptosis of 468H1047R. It was concluded that the latter was not significant, but I think that's something that needs to be a test that needs to be rerun, because even a slight increase in apoptosis should be of concern, especially when you're trying to convey the fact that these genetic modifications inhibit apoptosis and your data says otherwise, even if this otherwise is slightly above your margin or you consider it non-significant. Towards the end, they did discuss that previous studies reported that there was no association between PIK3CA mutations and the PAM pathway activation. So again, we would have to look at these studies and see their way of testing, but it's basically saying that another study kind of nulled their conclusion that the PIK3CA mutations and the PAM pathway were correlated. They did mention that there was no statistical difference and that the PIK3CA mutations led to poor prognosis, and that's something they mentioned in their discussion. I'm sorry. And this was due to clinical analysis populations. They did mention that their population sample was too small, so I think it's erroneous to reach the conclusion that mutations and poor prognosis are linked when the sample they tested was definitely too small. They did mention that they must analyze patients with the same TNBC stage and chemotherapy studies, because they did mention that the patients they studied all underwent different types of chemotherapy, and that can yield different results because different drugs are gonna behave differently. So you can't make the conclusion that prognosis is gonna be low when you're not even testing the same drug. And the drugs their patients were using were not epirubicin, so it's not comparable to the data collection they were testing because they use epirubicin strictly. And they did conclude that epirubicin concentrations might have been too low, which technically it would have yielded different data. They said this because the tumor volume we mentioned previously did increase even after treatment, which is the point we were trying to make. We were trying to emphasize that the gene mutations were inhibiting successful chemotherapy treatment. Future works, they did mention that the PIK3CA could be used as a biomarker for chemotherapy resistance. So again, this is clearly an important gene when it comes to why some TNBC patients are resisting to chemotherapy. One second. They also mentioned that PI3K pathway inhibitors and AKT inhibitors are being used and to test their effect on reversing chemotherapy inhibitors. So in other words, they're testing whether blocking these pathways, specifically the PAM pathway by inhibiting the PI3K or AKT, if there's going to be an enhanced sensitivity to chemotherapy. But again, that's something for future work that we are excited to see. I would suggest that they repeat the experiment with higher concentrations of epirubicin, especially considering they said they wanted to see halted tumor growth, especially on the control group, because even their control group, it looked like it wasn't being, it wasn't showing chemotherapy sensitivity because those tumors still kept growing, and that's not something we expect to see in in a control group. We expect chemotherapy to work on the control group. Maybe they needed more time, or again, they needed higher concentrations of the chemotherapy drug. But they did mention that they are currently recruiting more TNBC patients for future studies, so that's exciting. I'm assuming this is going to take a long time, considering this was recently published and the patients they previously used were from patients from 2014 and 2016. So I'm sure it's going to be at least a couple years until we do see their findings with these patients. So in conclusion, I would like to thank Dr. Shawbaugh again for giving me the opportunity to present this to you guys. And if you guys have any questions, please let me know. Thank you all so much.